Good afternoon, everyone. Thanks for the introduction. So initially, when we started wanting to look at invertebrate venoms and muscle damage, initially our focus was on snake venoms in our lab with Shakti, and we wanted to see whether invertebrate venoms sort of impeded skeletal muscle regeneration in a similar manner to that of vipers, or whether proper regeneration was being seen. But also to look at them as potential sources for bioprospecting, as maybe potential like alternatives to CTX, or as another molecule that could be used for other diagnosis or research tools. Um, so when we're looking at skeletal muscle regeneration, the main processes that we're looking at start with the satellite cells. So we have these as cultured myoblasts. So ours are AB1190s, which are human skeletal myoblasts. From there, once injury occurs, these cells basically activate, they start to proliferate, then they migrate to the area. Then from here, they will differentiate and basically form myotubes. So with this in mind, our in vitro work is sort of focused on these main different aspects to see how the venoms impact these processes. Um, and the main areas that impede regeneration from viper bites is the damage to the vasculature, damage to the extracellular matrix, and then you get improper regeneration. Ischemia, again, from the lack of blood flow with the damaged vasculature. And then you also get improper differentiation or asymmetrical differentiation of the satellite cells. So you don't replenish the pool that is necessary for future recovery following damage. But yeah. So initially we did a screen of 30 different invertebrate venoms to try and identify some species that we would want to work with. The first three species are scorpion venoms, which we ended up using for a blood screen, which I won't talk about today. And then basically, so we initially screened on MTS, so we're looking at cell viability, and LDH, so cytotoxicity or pore formation. And from here, we basically selected A4, B10, and C4. So A4 is Aponopelma annex, B10 is Omophymus violaceps, and C4 is Selenocosmia javanensis. There is also A5, A6, and A7 that were of interest. They belong to the Brachypelma genus, but someone else in our lab was using those for a blood project, so we decided not to go with those. So Aponopelma annex, or the Texas tan tarantula, probably very unlikely that you've ever actually heard of it unless you're from the area. They're not medically significant. There's no bite statistics. I can't even tell you exactly what happens when someone bites, if one bites you. Trawling through different tarantula keeping sites, people just say that it really hurts, and that's about it. There's a little bit of swelling. But yeah, other than that, I couldn't tell you anything else. Same pretty much goes for Omophymus violaceps. It's a very pretty tarantula, so it's very popular amongst the pet trade. And these ones are actually quite aggressive in terms of tarantulas. So bites are more common, but still no statistics for envenomations, be that in the pet trade or in the wilds, and also no effects in literature in terms of the envenomation. And there's actually no literature available about the venom for either of these species or their effects. So first of all, we want to try and profile the venom. So initially just an SGS page. So you've got A4 and B10, which are annex and violaceps. They're the main ones I'm going to talk to you about today. They are predominantly low molecular weight. So you've got a load of peptides around the 10 kilodalton and below range. And then a little bit around the 37 to 40 kilodaltons. Other than that, there's nothing else there. So this is 50 micrograms of venom per well, but this is based on the concentration or the optical density told to us by our venom supplier. So this could be inaccurate, and it could probably suggest why B10 is significantly less visible than the others. I don't know, I couldn't tell you, but we basically proceeded assuming that his concentrations or amount of venom he supplied us was accurate. So following MTS, we wanted to do a fusion analysis. So Shakti mentioned this briefly yesterday, but essentially we grow the myoblasts and then we differentiate them in a differentiation media, causing them to form myotubes. And basically, we then try and look at how the coverage area is affected by the venoms. So therefore the viability and its ability to actually fuse together. So interestingly, though, with these two venoms, they are having very different effects. So Annex is pretty much destroying all the myotubes at around 15 micrograms per mil, whereas Violaceps were actually seeing an increase in the size of the myotubes and their fusion index. And this is at 15 and 7.5, 30 is only affecting the fusion index. But if we go any higher with Violaceps up to 60, then all of the myotubes are gone. They're either detached or they are no longer viable. So next was, so this is what we'd also call an atrophy assay. So it's very similar to the fusion assay, except that we differentiate the cells and basically form myotubes first before we add any venoms to them, basically exposing them to the venom for only one day. Okay, I've put the wrong logo, but okay. And we are basically seeing a very similar effect. It's not quite as strong because they're not exposed to the venom for as long, but we are still seeing a loss or a lack of viability in the myotubes at 15 with Annex, and we're seeing an increase in the coverage at 15 microgram per mil, but at 30, we're actually seeing that they are no longer viable. Okay. So following this, we wanted to see whether we could see the same effect in vivo. So we injected 60 micrograms of each venom into the right tibialis anterior of a mouse. And we basically tracked, or not tracked, we dissected the animals at day 1, 3, 7, 14, and 21. These time points sort of tie in with different processes of regeneration. So day 1, we just wanted to see the acute damage and see what effect the venom has before any regeneration starts to occur. Day 3, we expect to see some form of inflammation and the start of regeneration. Day 7, the fibers should be centrally located. So you'll actually see regeneration has already started to occur. And by day 14, it's possible that the regeneration could be complete. So in the cases of CTX, we usually see complete regeneration of muscle fibers by day 14. We extended this to day 21 because we didn't see full regeneration in the preliminary screening that we did. In the graph here on the top, we're basically tracking the changes in weight of the TAs. We're comparing basically the right TA that's been injected to the TA of the other animals. And we're actually seeing an increase in the weight of the treated muscle by day 21. It's not significant. It was in the preliminary testing that we did, but it is still an increase nonetheless. So following that, we wanted to try and analyze the muscle sections and sort of get a better picture of what's happening to them. So I know you can't really see this, so I have put slightly more zoomed in ones so that you can hopefully see a little better. So the control is pretty much consistent throughout the 21 days. So I'm only going to show you for day 1 so you can get a better image of the future ones. But we're basically starting to see all the purple dots. They're all the nuclei. So you're actually seeing a lot of damage to the fibers already. And it's quite extensive. It does cover a large proportion of the muscle, which isn't something we typically see with a lot of the snake venoms. Maybe it's because we've put a lot more venom in, or it could be that it's better at spreading through the muscle than a lot of the venoms that we're using, but I don't know. So yeah, that's day one. Day three, we are still seeing some very extensive damage, especially from violiceps. We're pretty much half the screen cleared with no fibers present. Annex, not quite as extensive, but we are also seeing a lot of infiltration. So we're starting to get an immune response that's starting to come in, and hence why we saw that increase in weight by day three. So day seven, we're starting to see the fibers are nicely centrally located, especially in the terms of violiceps. So we're actually seeing that regeneration is actually occurring quite nicely at this point. It seems to be on track, and the same thing with annex. Not quite as much though. And then by day 14, all of the fibers are centrally located. They are relatively homogeneous in size. There's definitely still some larger ones and some smaller ones, but they are starting to get back to normal size and normal physiology that we'd expect. And by day 21, they look very similar to the control muscles. Very nice homogeneous size. There's very little infiltration left, and nice regeneration has occurred. And then to make sure that the increase in weight that we observed wasn't due to fibrosis, we did a Pixius red staining, and we've basically found that day 14 and day 21, there is no significant sign of fibrosis from either species. And annex actually has less fibrosis than the control at day 14, but that's likely just due to the differences between the animals, not due to the venom. Yeah, so far, and then, sorry, we did some blood biomarkers. So when we dissected the mice, we also did cardiac puncture and collected the serum and then sent that off for analysis. So we see very elevated creatine kinase levels at day one with Omophilus violiceps, and elevated LDH and general protein markers, mainly from violiceps. The rest don't seem to be having a huge effect. And then some of the issues that we've faced so far is that the venom is very expensive for a venom. It's £85 a milligram, which is a lot more expensive than snake venoms that we use. Very limited availability. We've only managed to get 10 milligrams of each venom so far, and the venom supply has gone into liquidation. So we're now struggling to find more venoms. We can't find any animals to replace them. So we need to try and figure out what we do next. If we want to try and isolate toxins or peptides, how do we do that? We don't know yet. Yeah, so that's sort of the issues that we're facing at the moment and sort of what we need to try and work around for the rest of the project. Yeah. Thank you very much for listening, and thanks to my supervisors, Jose, and the ITTP for funding this project. Any questions today?